عليكم طلابنا الأعزاء. اليوم المختبر الثاني What is mitosis؟ أي الانقسام الخلوي. Mitosis is a type of cell division in which one cell, the mother, divides to produce two new cells, the daughters, that are genetically identical to itself. In the context of cell cycle, mitosis is a part of the division process in which the DNA of the cell's nucleus is split into equal sets of chromosomes. You can see on this black figure here over here, cytokinases. We got prophase, metaphase, anaphase, and telophase. On this slide, let's start by looking at the cell right before it's begun to its mitosis. So we have here the whole cells, chromosomes, nucleus, DNA, and nuclear membrane. Prophase. In the early prophase, the cell starts to break down some structures and build others up, setting the stage of division of the chromosomes. The chromatin condenses to chromosomes. The meiotic spindle began to form. The spindle, what is spindle? It's over here. So the spindle is a structure that's made up of microtubules over here. The microtubules, as you can see, where's the arrow? Spindle is a structure made of microtubules. Spindle grows between the centromeres as they move apart. In the later prophase, sometimes also called the prometaphase, the meiotic spindle begins to capture and recognize the chromosomes. I think that's easy to interpret it in the figure over here. The late prophase or pre-metaphase, the nuclear envelope breakdown and the chromosome fully condensed. See the next slide. What is spindle? So we have the spindle anatomy over here. We got the ester over here over the top and kinetochore microtubules, and we magnified the centromere over here, and we got the kinetochore over there and the tubulus. The metaphase. In the metaphase, the spindle has captured all the chromosomes and lined them up at the middle of the cell, ready to divide. All chromosomes align at the metaphase plate. You can see here in this figure, chromosome lined up at the metaphase plate in the middle. Anaphase. In the anaphase, the sister chromatids separate from each other and pull toward opposite ends of the cell. Kinetochore microtubules pull chromosome toward the poles. The poles يعني قطب إلى الأقطاب الأعلى والأسفل. Microtubules push poles apart تدفعها إلى كل جانب الأعلى والأسفل. Telophase. In the telophase, the cell is nearly done dividing and it starts to re-establish its normal structure. The cytokinases, division of the cell contents, take place. The meiotic spindle is broken down into its building blocks. Two new nuclei form, one of each set of chromosomes. Nuclear membranes and nucleoli reappear. The chromosomes began to decondense and return to their stringy form. You can see in the telophase, chromosomes start to recondense and spindle disappear, nuclear membrane reforms, and nuclear reappear. Cytokinesis. In the cytokinases, the division of the cytoplasm to form two new cells overlaps with the final stage of mitosis. It may start in the either anaphase or telophase, depending on the cell, and finishes shortly after telophase. You can see it's in both differences between cell and plant. Animal cell and plant cell. On this slide, you can see the whole process. Intraphase, prophase, metaphase, anaphase, and telophase kinases at the end. Thank you very much for watching.